So good morning, good afternoon, everybody. Welcome to this webinar titled Australia's Leading Role in Countering COVID-19 through Global Research Collaboration. My name is Anne Green. I'm Chair of the New South Wales Division of the Australian Academy of Technology and Engineering, known as FTSE, and I'm delighted to welcome you all to this function, which I'm sure you're going to find fascinating and informative. So in the spirit of reconciliation, I'll begin by acknowledging the traditional custodians of country throughout Australia. We pay our respects to elders past, present and emerging, and extend that respect to all Aboriginal and Torres Strait Islander peoples today. As we share our own knowledge, I acknowledge the most ancient and enduring knowledge in the world, embedded forever in custodianship of country. So we're joined today by fellows of FTSE, members of the Union University and Schools Club, as well as friends, collaborators and partners from across Australia and internationally. So welcome all. FTSE is a learned academy of independent experts, and we help Australians understand and use technology to solve complex problems. So our fellows are Australia's leaders in applied science, technology and engineering, and we bring them together to provide impartial, practical and evidence-based advice on how to achieve sustainable solutions and advance prosperity. So the logistics for today's webinar, technical organisations via the Secretariat of FTSE, and I thank them for their assistance. All participants will have their video and audio muted centrally. However, there will be ample time for questions and comments after the presentations, and please use the Q&A function for your questions and they will be passed then to the panel. And also for your information, the webinar is being recorded and the video will shortly be placed on the FTSE website. So for today's webinar, we have been extremely fortunate to secure 3 eminent Australian scientists to explore the pandemic and how it has changed the research landscape. Australian scientists have had a profound role in understanding the dangers of viruses like COVID-19. They are increasingly recognised for their vital contributions to collaborative research with other leading scientists, including those from the USA, UK and China. So I'd now like to introduce the first of today's panellists. First speaker to set the scene and the context will be Professor Stephen Simpson, so the Executive Director of Obesity Australia, the Academic Director of Charles Perkins Centre and Professor in the School of Life and Environmental Sciences at the University of Sydney. Steve has had an extremely broad research career. He returned to Australia after many years in Oxford. He is co-author of the integrative modelling framework for nutrition called the Geometric Framework, which is applicable to a wide range of organisms from slime moulds to humans, and is one of the world's foremost entomologists and nutritional biologists. In 2009, he was awarded an Australian Research Council Laureate Fellowship and was named New South Wales Scientist of the Year. Steve has received numerous other national and international awards, including being elected a Fellow of the Royal Society London and the Australian Academy of Science. In 2015, he was made a Companion of the Order of Australia for eminent service to biological and biomedical science. Steve will have more to say later, but I'd now like to introduce Steve Simpson.
Thank you so much, Anne, and a welcome to everybody. And please, may I also pay my respects to the traditional owners of the land upon which I'm sitting, the Gadigal people of the Eora Nation. It's fallen to me to introduce the broad themes, if you like, of what we're going to be discussing today. And as Anne said, they're really the lessons that have emerged out of how the research sector has responded to the COVID crisis. And for those of us in the research sector, and indeed I would venture those of us who perhaps are in government as well, there have been some really important lessons that have emerged. So these will be essentially the themes for the webinar today, and we'll illustrate them through our 3 more detailed discussions around particular aspects. But I thought I'd begin with just some reflections on what have for me been the critical lessons that we've learned. I think the first thing is that the dichotomy between pure discipline-based discovery research and applied mission-oriented research has turned out to be a false dichotomy. Rather, COVID's shown us that we need the capacity to convene experts rapidly, both nationally and, as Anne indicated, internationally, to come together to tackle complex problems. And that we were able to do that was because we had a research workforce that includes disciplinary experts that span a range of different disciplinary areas. More than that, though, who are adept at collaborating in multidisciplinary teams. If you're going to have a workforce, it's going to need to include not only, we've seen those in STEM disciplines like ourselves, but also those colleagues who are in the humanities and the social sciences. And next, another thing that's really struck me during this pandemic is that if we in future, as a research sector, are going to have the social license to operate, we need to be able to communicate with and listen to civil society. But in return, the other side of the bargain, we would argue, is that all levels of society need to come together to help tackle the big problems that face the world, not to ignore them or worse, subvert the evidence that they exist. So there's some of the, I think, bigger themes that will pervade the next hour or hour and a half, and we'll pick up on the story in relation to COVID with our next speaker, my friend and distinguished colleague, Professor Eddie Holmes. I won't go into his biography in great detail. Eddie has been at the very forefront of both warning of a pandemic and in understanding the evolution, the emergence, the origin of the COVID-19 pandemic. He too is a fellow of the Royal Society of London and of the Australian Academy of Science. He's at the University of Sydney, the Charles Perkins Centre, and in the Mary Bashir Institute, which works on infectious disease. So with that, I'll hand to Eddie.
Thank you, Steve. I'll just share my screen. Right, good morning, everyone. Oops. So what I want to do today is really just give you, I've only got 6 slides, a very brief just overview on how we discovered this new virus, the virus that's causing COVID-19, and a bit about how for the future we may try and stop these catastrophic events happening again. I think that, and what I want to emphasize just today very briefly is how the way we discovered it, and for the future, we have to have at the very heart of that international collaboration. Everything will fail unless we globally act together. So that's the kind of overall theme, what you're going to hear just in a few slides. So I'm sure you would have now seen, as these are very common on news outlets and on the web, these kind of maps of the world showing over in recent years the number of these what are called emerging infections. So emerging infection is just one that's appeared recently or has increased in prevalence or geographic range recently. And this whole science really started in the early 1980s, so 40 years ago, basically, once we discovered HIV. And since that time, we've documented the kind of emergence and spread of these novel pathogens globally. And you can see here we have things that I'm sure you're very familiar with, like yellow fever or Hendra, of course, in Australia, SARS in 2003. And since this map was drawn, of course, we've had Ebola in West Africa, we've had Zika in Brazil, and of course now we have COVID-19 coming from Wuhan. And so you can see this is a global picture, and so clearly that tells you that a global response is completely integral to understanding this. And the basis to all these emergence events, and I've written it up there for you, is host jumping of pathogens. In other words, what these diseases are, what they all represent, is a pathogen, particularly a virus that I work on, jumping from another animal host into a human. And the way humans live today, the way there's changing land use, deforestation, the increased urbanization, the mega cities we live in, global travel, all those things have just fueled these huge number of emergence events to occur, COVID-19 being, of course, the most recent and most devastating. And they will occur in the future. This is just the way humans live. It will happen. So because of this problem, what many people have now started to, including myself, have tried to establish are efficient ways to try and document very rapidly, if we do get an emergence event, what microbe is responsible for it? How is it spreading? And the way we do that is using modern genomic technologies. We take a sample and we can sequence the genome of the pathogen extremely quickly. And so, so quickly, in fact, we can call it actually real time. So as the outbreak is occurring, we can actually find out what it is and track it. We published in a Nature journal that last year, feels like a long time ago, just a kind of description of how this might be done. So first step would be is you detect a new pathogen, you get your first cases here, someone who's sick, and you can use various bits of technology to find out what that pathogen causing the illness is. Now, just to show you how quickly things have moved on, it took about 10 years, it took about 2 years to find that HIV was the cause of AIDS. And for SARS-CoV-2, COVID-19, took us about, a week or so to work out what the pathogen was. That's now done very quickly. Once we have that, we can try to find out where it's come from, what the animal reservoirs are, and I'll discuss briefly and shortly. We know that bat viruses are very close to the one that's spread in humans. We can start to follow chains of transmission. Of course, you're now looking at the outbreaks in New South Wales, particularly in Victoria. And this genomic technology is allowing us to work out how the virus is spreading, what the clusters are. So we know, for example, using genomics, that all the cases we're seeing in New South Wales, they're all derived from the outbreak in Victoria. And genomics has been really integral to doing that. And then we can start to look also at the kind of global spread of the virus. We know, for example, in Australia, how the virus came in early on and how it spread. So all those techniques are available. And my group particularly work at the kind of front end here, the first thing, trying to work out what viruses are jumping from animals to humans. And with that information, we could try and develop ways to stop that happening. So what we do is we sample between places where humans and animals interact. And I like to think of those as fault lines in emergence. They're kind of stress points where pathogens can jump host species. We sample livestock, domestic animals, wildlife, where they interact with humans to see what's moving. And we, for example, I do a lot of work in China, I'll discuss it more in a second, and we can sample, this is in Southeast China, we sample rodents, particularly around people's homes, rodents and insectivores as well. And these carry, if you sequence them, you do these technologies here, they carry lots of viruses that can jump, sometimes do jump, and sometimes can cause very nasty diseases. So we sample a lot of things, they're called bunya virus. I won't discuss what these are. It's the name of a virus we find. And these bunya viruses that often passed on by ticks can cause very nasty hemorrhagic fevers. Here's a patient here from China showing the kind of classic manifestation of hemorrhagic disease. If that kind of capillary leakage occurs in internal organs, it can be fatal. Luckily, these viruses don't spread very far in humans. We can kind of stop them, unlike COVID-19. So that's the kind of work that we do. And then just to give you a very particular example of how we got to where we are with SARS-CoV-2. For a number of years now, I and my colleagues have been sampling animals in China. So we go to these photographs now taken on my phone. This is from a village in southeastern China. You can see stepped sort of rice cultivation there. And there are caves they take you to. So kind of this is inside a cave, it's kind of in a chamber in a cave. So then you put your PPE on. Hopefully now I'd wear better, this is me, I'd hopefully wear better PPE than this nowadays. And you go in these caves and they contain bats, okay? And we sample these bats and we sequence them. We do these technologies, look at these genome sequences, and we find a whole bunch of viruses, including coronaviruses. So there's a paper we published a few years ago with some Chinese colleagues, and we discovered a whole host of these bat coronaviruses, some of which are very closely related to the virus that's now spreading in humans. And we wrote at the time that basically that these coronaviruses we're seeing in bats, that some of those will very likely emerge in humans and be a public health threat. And I wrote this, and many of my colleagues who do the same work, we saw these things happening. We saw these bat coronaviruses, and we could see they were jumping species boundaries, and we knew this was a problem. So this is the idea this was going to happen was out there, and people had been warned. It was obviously at some point this was going to happen. So then, winding forward to what happened at the end of last year, what happened? I'm sure some of you may have seen. There's a website, a very good website, which you all very good thing to look at, called ProMed, and ProMed gives literally daily reports on diseases emerging in humans and other animals. So here was a ProMed report from the end of last year, and it just basically says, this is the first thing that I saw. It said there were 4 cases of pneumonia in patients from Wuhan linked to a seafood market there. Now, I actually had colleagues in Wuhan. Lots of the sampling that I do in China is around Wuhan. We had a very big respiratory disease study going on in Wuhan at the time. And so here's this picture here. This is Wuhan Central Hospital that became famous in the news. Lots of the patients turned up. And you can see my name there. This is 2016. So I gave a talk at the hospital to set up the project that we did to sample people with respiratory disease. So when I saw this report, I thought, oh my gosh, this is right where we are working. I need to get involved. So I contacted my colleagues in China, and they were already working on the samples. And within a few days of this report, in fact, January the 5th, so less than a week after, my good friend, Professor Zhongzhen Zhang, had sequenced the complete genome. We then, on January the 5th, we informed the Ministry of Health in China that we had a virus, it was a coronavirus. We thought it could be respiratory. And we also sent it to the US, what's called GenBank. It's basically the global repository of genome sequences for everything. So it was sent there on the same day it was sequenced. Okay, really amazing, important, the same day, just so everyone could see it. And then a few days later, I posted that genome sequence. This is the first release of the virus anywhere in the world, on a website called Serological, and I tweeted about it saying, here's the new genome sequence. And the great thing was, within a week of this sequence being made available to everyone in the world, scientists in Germany had developed a PCR test to track that infection in people, and that's still the test used today. So remarkably quick, within 2 weeks basically of knowing about the disease, we had a test because the genome was sequenced and released. So that's a hugely important, just a case of how important it is to kind of share data and work globally. So that's what happened. So then you can all see the consequences of what's happening to us now. I don't need to labor that, but the question is, how are we going to stop this ever happening again? And I think there are 3 things that we can try and to do, all of which will require global frameworks to work together. The first thing is that this virus is about, while I was in China, about our exposure to wildlife, how humans have changed their interaction with the natural world because of deforestation, changing land use, things like the wildlife trade, wet markets, zoning, all those things have changed. So somehow we have to rethink our relationship to the natural world to kind of get physical distance to the animals that carry these infections. They do, and we will get them again. So for example, this is a picture I took from this is actually from my phone, from that very famous market in Wuhan. I don't think that market is actually now the source of the infection, but still, this is a raccoon dog for sale. And there are lots of these animals for sale, these mammals in these markets, and these animals will carry viruses. So at some point, we have to just stop these practices or limit them or control them or regulate them, because these things will happen again. We must have a really much better global surveillance network going. So some countries have built them. Australia, I think, could do better. I'll discuss maybe at the end of this talk. And that surveillance can be genomic, these are the tools I've said at the start there, or serological. So you take people's blood and look at what past infections they've had. It can involve social media. All those things need to be much better harmonized. This is a picture here. This is a ProMed map, just showing on a particular day the alerts that they're putting out there for the world to look at. Look, here's the infections here, watch these. This chap here, I took into a picture 2013, it's from a village in southeastern China. And he's a local China disease control expert based in that single village. And he's written on the kind of village notice board here, and it's saying, look, there's an outbreak of avian flu in the population, watch out for chickens, they could carry it. So that's very local level community engagement, the kind of national thing. We have to integrate all these things with new high-tech tools to monitor infections. That has to happen. And finally, the kind of Apollo project, the whole thing, is we have to try and develop and stockpile now antivirals and vaccines that can broadly recognize a whole range of pathogens. Of course, now the whole world is waiting for a vaccine to come, and it may come next year, it may come the year after, it may never come, who knows. We have to try and have those ready before we go, rather than having to wait, put our lives on hold for years waiting to happen. When SARS came out in 2013, there was an initial flurry of people looking at vaccine research. That then ended, the funded ended very quickly. We have to continue. We have to try and make these universal vaccines and antivirals now that we can stockpile from the future. That's very much easier said than done. We've yet to make a universal flu vaccine despite many years of effort, but we have to kind of research on this. Because if we have those things in place, we can really try and stop these devastating things happening again. So, and again, all these things are going to be hugely collaborative and globally collaborative. We can't exclude any individual countries in this. It has to be a global effort. So I think I'll end there. I hope that's going to set the scene, and perhaps we can have questions later on.
Thanks very much, Eddie. Now, if you unshare your screen, I'll share mine. Okay. All right, so I'm going to pick up on a related aspect that's emerged out of the COVID experience, and I'm going to begin with a data slide that comes from the UK, and it's pretty terrifying actually in several respects, and is really an important indicator of a problem that we should be taking much more seriously than we are. What this shows is case fatality rates for people hospitalized with COVID in the UK. And it's mirrored, the data are mirrored around the world. So this is just a fairly typical illustration. You'll see two things here. You'll see the histogram bars rising steeply as you go to the right, and that's indicating that your risk of dying is exponentially related to your age. The older you are, the greater the risk, if hospitalized, of your dying. But on top of that, obesity profoundly increases the risk as well, in particular during those middle years, sort of aged 50 to 65, 70. Now, the reason that's terrifying is twofold, really. One is that, and this is a success story, Australia, like other countries around the world, is an aging and indeed becoming an aged society. So this shows just the percentage of the total population aged greater than 65 and how that's changed over the years and is predicted to change demographically as we move into the future. We're getting older. Added to that, aging is the main risk factor for a whole suite of different chronic diseases. So here's just some of them shown here. This is the mortality rate per 100,000 for a range of some of the big chronic diseases: cancer, Alzheimer's, heart disease, and stroke. And you'll see that they all go up in lockstep with age. Accordingly, if you take data on hospital bed days or PBS medicine costs, they too go up steeply as we age. So that's an issue. That's an issue that we all know about in Australian society. But we're also an increasingly overweight society. So this map here, 2007, 2008 to 2011 and 12, you can see the growth in obesity across Australia. to the point now that 2/3 of adults and 1/4 of children are classified as being either overweight or obese. And that's particularly apparent when it comes to social inequality. So a higher proportion of adults in the lowest socioeconomic groups suffer obesity as distinct from the higher socioeconomic groups. And what this means is that the healthcare system is facing a looming catastrophe. We spend billions of dollars every year diagnosing and treating chronic diseases after they've occurred. And our colleagues in the US, they spend 91% of the 3.3 trillion that's spent on healthcare related to dealing with chronic disease burden. Even our Australian Treasury's intergenerational report in 2010 estimated a doubling of the percentage of GDP that's going to go to health by 2050. And the Committee for Economic Development of Australia warns that this simply is unsustainable. We need new models of healthcare. Now, why we're in that mess is again partly a success story. We've built a safe and sustainable food supply for our society, but in a sense, we've built a world that meets all of our ancestral heart's desires. So our food production and supply system is high in calories and high in the qualities that were missing ancestrally, and we've become designed to really desire things like high fat and sugar. We've got cities, homes, and workplaces that are designed to allow us to minimize our energy expenditure. We've got economic systems designed to value wealth over health. We buy things that we like rather than are good for us, and hence companies that sell us what we want to buy prosper in the economic landscape. And we know that prevention is better than cure, but it doesn't in the short term make much profit, nor indeed garner many votes. So what we've essentially done is taken an animal, ourselves, that evolved in a world which, like all other animals, meant that we didn't expend energy unless we had to, and we really valued certain properties about our food supply. And we've hacked that biology and our appetites. And here's a shameless plug for David Rabenheimer in my new book, which tells that very story. What we've essentially done is build a society in every respect that's kind of trapped us, and our likelihood of ending up sick in the modern environment is substantially greater than it need be as a result of that. But that's a fabulously complex problem to untangle, a difficult challenge to unweave. So what do we need to do in relation to the healthcare system? And what I'm going to argue, just very briefly, is that we need to consider moving from what you might argue currently is a sick care system to what's truly a healthcare system. So what we have now is very much a disease-centered approach to the way that we train our medical practitioners, the way that we research medical issues. So it's pretty well designed around the notion of developing specific pharmacological and surgical interventions for individual chronic diseases that allow you better to diagnose, treat, and control the symptoms of each disease. So drugs and surgery around particular organ systems. That's sort of the way, it's a caricature, I know, but still it's not a bad reflection of the way we do things now. What I think we now have the opportunity to do is to take a truly mechanism-based, prevention-focused approach, where rather than preventive policy, which is advisory and rather high level, instead we now have the basis for a deep understanding of the mechanisms that control chronic diseases and the environmental determinants and levers that can be engaged to actually prevent the development of these diseases before they happen. And the reason I say that is because what we're discovering, and this is Australian science, it's science internationally as well, is that many of the common chronic diseases, and they include mental stress, the diseases of sedentary lifestyles, excess calorie intake and unhealthy diets and more, they're all mediated via the same common metabolic substrates. And we can understand those substrates and their relationship to disease, and not just chronic disease, also to infectious disease. So the relationship between inflammatory systems and metabolic systems and obesity on the one hand and COVID on the other is becoming clearer and clearer. But the fact that there is this common underlying substrate means that you've got the capacity now to target with lifestyle interventions as well as drugs, but not only with drugs, these specific pathways and ways that give you the opportunity for a new medical approach to the primary and secondary prevention of chronic disease. So my challenge really is that we need to squash the curve, taking the metaphor from COVID, for chronic disease, and we need to use this new preventive health strategy to do that. Now, that happens to be exactly what we're doing at the Charles Perkins Centre at the University of Sydney, where we're bringing that sort of basic fundamental discovery science around the metabolic drivers of chronic diseases, the common substrates, how you can manipulate them through drugs and lifestyle, particularly diet, and how in the end you can understand their interconnections, not only within health, but also more broadly through their interactions across the whole of society. And I'll end here with an animation which gives the sort of genesis, if it's going to run, the genesis of the network of activity at the Charles Perkins Centre over recent years. Now, it doesn't look like it's going to run. It's probably too slow. But nonetheless, what it shows is an interconnected network of collaborative research, which has brought in practitioners from across disciplines to come together around issues which go from, as you can see here, the gut microbiome, all the way through to the use of technology, avatars and apps to change people's behavior in relation to their health, to change the food system, and much, much more. So I think on that note, I'll end and hand over to my next esteemed colleague, Professor Ben Eggleton, who is the director of the Sydney Nano Institute, another of the multidisciplinary initiatives at the University of Sydney. Ben is another extraordinarily distinguished scientist. He's one of our most notable scientists working in areas that span the work of FTSE and the Australian Academy of Science. He's a fellow of both, and Ben's going to pick up on the technologies that may take us into the future post-COVID.
Thanks, Steve. Okay, so you've stopped sharing, and I'm going to share. And let me just make sure that's on the slideshow. And you can see that slide. Yes, and you can hear me. Great. Thank you, Steve. And it's great to be with you today with such distinguished company. So as Steve says, I'm going to pick up and give a perspective from the University of Sydney Nano Institute, but also with my other hat as director or co-director of the New South Wales Smart Sensing Network. And so you can actually see the virtual background behind me. I'm not actually in the clean room downstairs, but that's the ground floor of the Sydney Nanoscience Hub, which is the headquarters for the University of Sydney Nano Institute. As Steve said, we are one of 8 of the university's flagship multidisciplinary initiatives. Our sister institute is the Charles Perkins Centre, the Brain and Mind. You're familiar with those, and there are others. Sydney Nano, anchored in science, engineering and medicine, but has very good links into the arts and social sciences, the School of Faculty of Business, Architecture, Design and Planning, and even the Conservatorium of Music. We're a relatively new institute, so we're still very much infant in terms of our long-term journey. I've been in the role for 2 years, and I'm going to give you an example in the next 10-15 minutes of one of the exciting initiatives that has indeed responded to the COVID-19, and I think it really exemplifies our ability to bring together across disciplines new opportunities that address some of the grand challenges, really to pick up on Steve's narrative at the beginning. So Sydney Nano, although initially anchored in the physical sciences, its research framework really can be broken down into two major thematic areas. On the one hand, very strong programs in material science, nanomaterials, and at the same time in the life sciences, which obviously is the focus of the conversation we're having today. Our flagship project are the so-called Grand Challenge projects that I launched earlier in 2019. And you can see on the slide the 6 Grand Challenge projects. These are really projects that are multidisciplinary, pan-university, mission-focused research projects that address some of the really important global issues facing humanity. But I'm going to take you on a relatively more recent journey, which is part of an initiative I launched early in 2020, which was to enable a nano-health strategy at the University of Sydney. And this is a collaboration between Sydney Nano and the Faculty of Medicine and Health. I worked very closely with the Deputy Executive Dean, Professor Mark Rees, on this strategy. And we've put together a framework that has at its base really strong capability, foundations, expertise, infrastructure in a number of key areas in nano-health. I'm not going to spend a lot of time, but you can kind of recognize the basic underpinning science and technology in nano-health, which is nanoparticle, computational nanomedicine, nanoscale biomaterial. And these research programs very much address those traditional challenges in health and medicine. Now, what I want to focus on in this conversation is very much around infectious disease. We've worked very closely with the Mehri Bashir Institute that Eddie is associated with, in particular Professor Tanya Sorrell, who's been part of this conversation. But I want to focus very much on the sensor theme.
Could you go full screen?
Isn't that full screen?
No.
In which case I will stop sharing and I will go back, and now I will. Is that right? Yes? Thumbs up.
Yes.
Everyone's happy.
Yes.
Okay. Right. So thank you, Anne. So let's have a look. So this is very much a multidisciplinary approach, as I mentioned, and we've been able to bring together researchers across science, engineering, and medicine with clinicians, pathologists, clinicians in infectious disease, and we're using co-design to really facilitate the prioritization. And I'm going to give you some more examples of what that looks like and what that means. So this is the world we live in at the moment, of course, is all virtual. Arguably, we're more efficient in this virtual manner than we might have been in physical meetings, but this is a snapshot of a recent gathering of my colleagues, and we've got representation here from science, engineering, medicine, and health. We've got representation from the business office, from the Knowledge Hub, from the School of Business. So really fantastic team of researchers and professional staff working together. So let me just pause. So I talked about sensors and the New South Wales Smart Sensing Network. So just to tease out specifically what we're focusing on here, of course, is smart sensors. Now, the FTSE community, very familiar with smart sensors. IEEE defines a smart sensor as essentially a combination of sensor and processing with a dedicated architecture to aggregate external sensor data. And our homes are now incorporating smart sensors. We're all familiar with smart sensors in the context of smartphones, the Internet of Things, big data, e-health, wearables, I'll talk about that, smart cities, smart farms. We're doing work in smart agriculture, smart devices. We're even doing work in smart defense, but let's not get bogged down on that today. So at Sydney, we have really two different categories of sensors that we focus on. On the one hand, sensors that directly detect the virus. So this is really the domain of Eddie. And this is pretty well-established technology. If you walk down the road to the RPA and you feel stiffly, you'll walk into a room, you'll fill out some paperwork, and someone will do a test. And within 24 hours, 36 hours, you will be told by a text message whether you're infected or not. We also understand the narrative around antibodies. We know that we generate antibodies, we can detect antibodies. Probably it's fair to say that detecting antibodies is somewhat easier than detecting the virus. But of course, the antibodies don't say that you're infected. They imply that you've been infected. And as I've learned from talking to my colleagues in infectious disease, the whole narrative around antibody detection itself is very complicated. There are different types of antibodies and they tell you different types of things. So we've got on the one hand, this whole conversation around integrating sensors into the smartphone, building sensors that might allow you at home to test yourself every day, but that's a kind of a way off. So we've been focusing on a slightly different question, which is using sensors to detect the physiological signatures of infection. So this is really in the context of the current situation where we'd like to be able to test ourselves pretty frequently. I mean, how often do we need to be tested? How do you manage this current crisis? At the moment, you wake up, you've got a sore throat. If you're a good citizen, you might walk down the road, get tested, wait 3 or 4 days, you're off a week's work. How do you know, in fact, that you need to get tested? Can we use sensor technologies to empower someone, inform them that they need to be tested? How do we protect hospitals from, so there's a whole bunch of questions that I'll talk to around sensors that can detect the physiological signatures of infection. And let's sort of broaden this out to say, well, actually, what we're really talking about is sensor fusion. So the idea of sensor fusion, which is really at the heart of the research I do in my group, which is primarily sponsored by the defense, it's the idea of using multiple sensors, different sensors, that when combined provide a more complete, more comprehensive and more accurate perspective on the situation that it's monitoring. Wearable devices that, for example, can continuously gather your temperature, heart rate, blood oxygen levels, might allow, using machine learning, using AI, to inform someone whether they're infected. Are there signatures? This is a question, this is a hypothesis that we're asking. Now, if you Google, you'll find that there are companies in the US that already claim to have smart watches that will tell you. There's a bit of a question mark there. We can have a conversation about whether that's actually feasible, whether they've actually got the right training data. So we've broken this down into 5 very specific problem statements that we'll be looking at over the next period as part of the co-design that I talked about that brings together clinicians, industry, patients, public health professionals, the LHDs, researchers, innovators. There are really 5 specific questions. The one that I focused on, I alluded to earlier, detecting infection in the home to inform testing. Relatively straightforward. Can we listen to someone's snoring pattern? Do they, when they're asleep at night, make certain noises that reveal infection? How do we improve screening at high-risk facilities? At the moment, we use remote sensing to detect someone's temperature. Well, we know that pretty well doesn't work, but we know that more sophisticated remote sensing might look at someone's face and actually, using machine learning, using AI, actually will know if someone's infected based on more subtle, more nuanced aspects of the response, for example, as they walk from a cold outside to a warm inside. How do we monitor during the acute disease phase? This is either in the hospital, in the home, or it could be in a hotel, using wearables, using other sensors. And then, of course, once you've been discharged and you're no longer infected and you're sent home, the end of the journey, it's not the end of the journey. It turns out there are a number of documented respiratory, neurological disorders that need to be monitored. And then you could take these 4 questions and plant them to a developing country and ask the question, well, this is all fine. This is all going to work in the context of a very advanced country. How do we deploy this in a developing country when they have none of the infrastructure? So just to wrap up, I mean, this is obviously a conversation that's been had around the world. Company Eggleton, some might have been familiar with, already are in the business of using technology. They have already put out in the marketplace something along these lines. FTSE claims when you check in, they're going to use some very basic sensor fusion to determine whether you're infected or not. We also hear about sniffer dogs. This is a lot of fun. Looking at sewage testing. And then there's this fascinating idea that's appeared in the newspapers, surveillance from above, using drones to monitor people. And this actually says, and I'm not sure how serious this is, but I put this out there to be a bit sort of provocative. They're talking about using drones to monitor the temperature, heart and respiratory rates, as well as sneezing and coughing. It's a little bit scary, isn't it? So anyway, that's sort of my perspective. And the last comment is just to say, globally, there's a massive nano community around the world, and we're in contact with a number of them. I'd love to have that conversation about how we leverage those global partnerships. Thanks very much.
So thanks very much, Ben. And thanks to all our speakers. Now I think we're going to move to some questions. So if you could put your questions in the Q&A box, but I already have some which have been loaded. So the first one I was going to ask was to Steve. You and Eddie both have a background in evolutionary biology. How important has that been for your work in health and infectious diseases?
Well, fundamentally important, and certainly Eddie can speak to that, because everything Eddie does is predicated on evolutionary biology. And I think, Eddie, you were the pioneer of the methodologies that actually allow us to track molecular phylogenies, the family trees of viruses. So
in this particular case, sorry, Steve. So in this particular case, emergence, so the fact that when we acquire a novel virus like SARS-CoV-2, that is an evolutionary process. The virus has to adapt to spread, to infect human cells and spread through human populations. That in essence is an entirely evolutionary question. Also, as Steve alluded to, the techniques that we use to track the spread of the virus, and genomics is absolutely now critical to try and track how the virus is spreading through our population. So we can link, in Melbourne, we can basically, link the current outbreak to a very small number of escapes from hotel quarantine. And that comes from genomics has been critical in doing that. So, and the way you analyze those genomic data is by looking at their evolutionary relationships. In exactly the same way as you might look at do an evolution of, of apes and whatever. It's exactly the same technology is used on that. It's just on fast forward, right? It's just going, this is virus. So evolution, because it gives you those comparative tools, is absolutely critical to the way that I think about this and many people think about this.
And certainly in the practice of nutritional biology, which is the area that I have a particular interest in, again, evolutionary principles have been foundational. So if you want to understand the current nutritional dilemma, all the pandemic of things, literally, that are associated with our relationship with diet, you need to understand the evolution of our biological regulatory systems, our appetites and their interactions with our food environments, to see where they've gone wrong and where we need to reinstitute certain principles in our environment to make our evolved systems work again, to help us towards healthy diets. And the best way of doing that is to survey the rest of the natural world and understand how other systems have managed to get it right. So if you want to understand the solution to a really complex problem, go out into the natural world and see how evolution has solved it. Therein lies the inspiration for understanding the human dilemma. And that really fundamentally has changed everything that we do and have done. And even more than that, an instantiation of exactly that set of ideas is the way we've developed the Charles Perkins Centre. We've set it up as a complex adaptive system to bring people together to start to innovate and to build upon their innovations and develop them using that same set of principles. You come up with new things, you infect other minds with your ideas. Where they work, they'll be promulgated. When they're not, then they'll die out. And you can generate extraordinary complexity and solutions to complex problems in ways you couldn't imagine. And actually, Ben, that's the same set of principles that are used in artificial intelligence and machine learning for trying to understand how to fuse data sets from sensors. It's the same principles.
Fine, so let me move on. I've got a question which will go to Ben. And someone has asked, could sensors be used to monitor change in the usual cognitive processing, so to indicate the impact of illness? And that also, can you then use that to identify individuals who are asymptomatic in this present COVID-19 pandemic?
Yeah, great question. I mean, is that is part B related to part A? Because part B is in itself a question, which is really at the core of what we're trying to do. Can you use a sensor to detect if someone's infected, even if they're asymptomatic? So what we know, in my understanding, asymptomatic doesn't mean they're not ill. It just means they don't know they're ill. I mean, I looked at Eddie. You agree with that statement?
So yeah, absolutely critical point this. So the reason why this virus is so much worse than the first SARS virus is that people are transmitting when they're asymptomatic. So the virus is absolutely replicating a high titer, but they have no symptoms. And that's your worst case scenario for any pathogen, because it's basically, you've got the silent spreader. The first SARS outbreak, 2002-3, all the people were only infectious when they exhibited symptoms, and that made it much easier to control. In this case, lots of transmission is occurring in people who are not have symptoms. So they're the absolute critical ones we need to target, because they're the ones that are so hard to quarantine, because we just don't know they're ill.
So yes, so that's at the core of the issue, and the question we're asking is, can we use sensors when someone doesn't know they're infected, they don't have symptoms, to detect signatures of disease? And it's all about the training data. It's only going to work if we've got enough data that we can use machine learning to look for these signatures, these patterns. That's the hypothesis. And so there are clues, and we've been talking to people in respiratory, we talk to people in cardiovascular, we talk to people in cardiologists, and there are certainly clues, and I'm optimistic. I think in terms of cognitive processing, absolutely, that's an area, and we are talking to people, in fact, in the Charles Perkins, in that area. So that's where wearables might reveal behavioral changes, or sensors might actually provide signatures. I mean, again, it's a hypothesis, it's speculative. I think the challenge we have here at the moment is getting access to loads and loads of data so we can train these sensors to look for these signatures.
Great. So I guess one of the key questions for all of us who are not in the front line of research, what are the prospects for a vaccine in the next 18 months? So I don't know, that's probably one to Eddie, because if we don't have...
Yeah, thanks for that one.
Yeah, it's going to constrain a lot of activities.
I mean, we have 150 plus, I'm sure people follow the news, maybe 150 plus, I lost count, vaccines that are in development. Some are in different stages of clinical trial. Some are at quite advanced clinical trial. Okay, so it's stage 3, which means it's a full test in a naive population. Those trials will take a while to come through, and we need to do them very carefully, because we cannot in any way afford to license a vaccine that's in any way not safe. So that will take a while to come through. The data at the moment says that the vaccines that we have, there's a variety of different approaches being used globally, and we need to have as many irons in the fire as we possibly can. The data at the moment suggests that they're safe. In animals, there is definitely immunity. There appears to be potentially some protective immunity. So we are waiting with bated breath. My take on it, this is a completely personal opinion, so don't take this in any way as read, is that I suspect we will get a vaccine within the year. I'm hopeful. How protective that will be is a different question. So how long a vaccine will give you immunity for is a more complicated thing. So obviously the ideal vaccine will be a one-shot vaccine that will give you lifelong immunity. I suspect that might be out of reach. We might be more into the influenza side of things. We have a vaccine that we need to continually repeat every year or so because of evolution in the virus. But I'm hopeful that we will get something. The results so far are encouraging, but we just need to go through the trials, and that's critical.
Wow. So we have another question, which again to all of you is, how do we sustain international research collaborations when we have all these travel restrictions? Now I know we have online collaborations and they can be utilized. Is that as good, or what are the restrictions for that? And then there's a corollary to that. What policy mechanisms can we put in place to keep promoting international collaboration? Maybe Steve, you could pick that first.
Is it as good? Well, when you reflect on the really important serendipitous things that happened over your own career, that happened at conferences where you were face to face, usually somewhere in a bar late at night after the formal events of the day, it's really hard to replicate that. And I think that is a challenge, trying to replicate the face-to-face unintended outcomes that actually spark some of the most exciting things that we all do. But, you know, as a first effort, occasions like this and get-togethers through Zoom and similar platforms are working, I think, a lot better than I expected that they might. So all is not lost. And we are always globally connected as scientists. We're always sharing data. We're always sending each other emails on social media platforms and the like. That can continue, and that eases some of the burden, that's for sure. What do we do in terms of policy settings? I think we need a real change in mindset. There seems to me a tendency to think about, and people even ask the question, do we need a local research industry when the big engines of research are overseas? Why don't we just buy in IP from overseas? That's the way of thinking about research as if it was a local car industry, which denies the essence of research. And that is that by being globally connected, you have the opportunity to play with the big boys and girls, and to have an influence and have a voice in a global community, which gives you the chance, if you've got a clever enough population, to have greater influence than your investment as a nation in the research that gives rise to those international players. We should be looking at our research budgets and our research enterprise in those terms. How can we leverage the cleverness of Australian scientists such that we can be heard across the world and influence the conversation across the world in ways that will punch above our weight? We already do that, but I think despite the system rather than encouraged by it.
So I have another question that says, how good is the modelling for the future incidence of infection? So if we look at the papers, of course, there's very alarming predictions about what's going to happen in Victoria. So I guess it's similar to the criticism sometimes on climate change, but the question here is, how good are we at predicting what's going to happen with our infections for COVID-19?
Shall I have a go?
Yeah.
I think the modeling's pretty good. I mean, I think that the models are very well established, and I think they are generally pretty accurate. I think I mean, what's happened in this pandemic, all the modelers have formed like a global club, and they're continually sharing models across different countries and cross-checking with each other. So I'm pretty confident about how the models are. I don't think the models are the problem. I think the problem is unfortunately people need to adhere to the rules, and I think and it's about individual behaviour for the common good, unfortunately. And I think that, I mean, what you're seeing now, this is a global thing, is that second lockdowns are far harder to implement than first lockdowns. And it's difficult to control the pandemic if people are mixing and they're shedding virus. Unfortunately, that's just the way it is. So I'm very confident about the model. I don't think that's where the problem lies. It's a difficult thing where the state has to impose quite strong restrictions on people to try and prevent these things happening. I'm afraid that's where we are.
Well, as a sort of related question, does the panel believe we should stay with suppression strategy or try for elimination? And is elimination at all realistic? Eddie
Oh, thanks.
Yeah.
So just a very important point I should point out at the start, that what's happening now in Australia is not dependent on the fact that we went for a suppression policy rather than elimination policy. That's a very important point to make. Anyone would have had the same effect. And it's because we had effectively eliminated the virus, but we had a breach in quarantine. And that would have happened in any circumstance, okay? So that's the first thing so in a little way, it's a bit of a moot point to discuss this. I can see both sides of the argument, and I'm going to try and dodge the question a little bit, actually. What I would like to see, actually, is a broader national discussion about this. What I'm alarmed about more than anything else is not so much whether we go for suppression or elimination. There are arguments on both sides, but this discussion is being done through the media, and that really worries me. And I would like to see a broader panel of people involved in discussing how we should approach this, and that's my number one. I have my own opinions, but I think it needs to be aired. We have a proper forum where when it shouldn't be is essentially, to be blunt, a few talking heads getting on the media and shouting who can shout the loudest. And I think we need a really informed discussion of it. And I think we're not having that at the moment.
I guess leading on from that is a question that says, what is being done to ascertain what the principal mode of transmission is? So is it an aerosol, or is it mainly on surfaces? And if it's, I mean, we've already talked about droplets, but aerosol is a different strategy.
So I wrote a report for the Australian Academy of Science on surface transmission. And so clearly the virus does survive on surfaces. For a while, clearly some surfaces more than others. So interested readers should go to the Australian Academy of Science report we read on this. I don't think surface transmission is the main mode of transmission. It's certainly possible, but to me, all the evidence suggests that the vast majority of this transmission is respiratory and happens basically in people's homes or in their workplaces. That's the number one mode of transmission. Now, within respiratory, the question is how much of that is droplets? Okay, so they basically sneeze and we cough and there's a kind of ballistic trajectory the droplet takes, or how much of it is in very small water particles. When we talk, there's a kind of diffusion, and that debate is ongoing. I would suggest to you that there's increasing evidence that aerosol transmission does take place. It may only be a small proportion of the number of transmissions, but I do think there is evidence for aerosol transmission to some extent. So, and how we incorporate that into recommendations for control is the next question. And again, this is a global discussion. This is not an Australian thing. Every country in the world is basically grappling with the same issues.
Okay, Ben, can I get a question for you? You talked about sensors and wearables. So what's the ideal type of wearables, the location, and how will we do it? Are we all going to have implants or something? So could you talk about, for detecting symptoms particularly, what are the ideal wearables?
Yeah, that's the great question. That's the sort of $1 million question. And it depends on a number of things, what you actually want to detect. But I guess where we are converging, we have kind of a number of different narratives there. We're very interested in the notion of the hearable. The hearable, it turns out, a sensor that sits on your ear or in your ear, seems to be well adapted to detecting many of the physiological sort of biorhythms that reveal health. And it doesn't sort of interfere. It also might even allow you to measure sun exposure. That's something I've talked to Ian Hickey about for many years, the idea of sensors that are part of a circadian rhythm study. So the hearable, next week we have a distinguished seminar from John Rogers from Northwestern University, a name many will know, really legendary US scientist who's pioneered sort of biocompatible devices. He has a wearable that sits on the voice box here. It's a little patch that sits just on your Adam's apple. Published this in Nature Nanotechnology a few months ago. And that seems to be the sweet spot. It seems to be quite really impressive. I almost included that in my presentation. So it can basically measure almost anything to do with your physiological sort of condition in real time, connect to your smartphone, onto the cloud, AI, and that's the holy grail. So look, it's a good question. I don't know the answer. That's the conversation we're having with the co-design, and it's going to be interesting. And there's a lot happening. It does come back to the training data. I mean, it's one thing to have a sensor that can detect the physiological sort of rhythm, if you like, but you need to have the data that tells you what signatures to look for.
So what about data security with these kinds of devices?
Sure. I mean, that's an overarching issue that we're all kind of, that's a separate conversation, isn't it? So let's make sure, let's assume someone else is solving that problem.
Okay.
It's a good problem, important problem.
It is. So another question, probably for Steve, is there a problem, and do you see a problem with transparency as businesses and governments tried to avoid this blame game and financial repercussions?
Well, absence of transparency is anathema. It's a problem. Generally, I think, and the blame game is a natural instinct, and we just have to grow up and get past all that, I think. And similarly, the vested interests that have driven conversations around some of the other big challenges facing us, whether that be climate change or whether that be issues around chronic disease burden, or whether that be some of the bigger issues that face the world in other respects. And I think we just need to get to the point where we have to come together across society and admit that there are challenges, admit that there are structures which will impede coming to solutions, and we just have to manage those. The vested interests that pervade the way that society operates now are there, and we have to manage them. And transparency at all levels is going to be important in doing that.
So another question, very interesting. What lessons have been learnt from the Swedish approach to herd immunity? Should we be following that?
No. Next question. No, look, I'll give you a more informed answer. So early on, clearly, there was essentially an enormous global experiment in how best to deal with this outbreak. And different countries took different approaches. And a lot of them basically, some of them went for hard lockdowns, some accepted that the virus would never be eliminated, therefore it was better to just manage it rather than lockdown. And what you're seeing now are the kind of endpoints of that experiment. Also, obviously, there's now a global concern about the different countries' economies, and can we really suppress people for so long because of the economic damage that does. The Swedish approach has certainly is an interesting one. I mean, it's certainly got, if you look at the numbers, it has far more cases and far more deaths than its Scandinavian numbers. Its neighbours, sorry. Its economy has fared slightly better, but it's still also suffered some constriction. But I think the worst thing I think the thing that argues against it is that they did this on the assumption that they get herd immunity, this magic proportion of 75% of the population or so, which when it gets that level, you are effectively protective. And they are nowhere near that. I think they're about 10% at most infected in Sweden. So there's still a long way to go, as are basically all countries. So, and people say, oh, we should adopt the Swedish approach. It's all very well to say that, but unless your systems are very well governed, I think we could have ended up as bad as Northern Italy. Okay, so that's the other thing. Don't just think if we let go our lockdown, we'll end up like Sweden. It could be Lombardy, and Lombardy was the richest province in Italy and had horrendous scenes in February. Absolutely terrifying scenes. So it's not the case if we had less strict lockdowns, we'd be like Sweden. I think that's just wishful thinking. We don't know how it would be. In the US, we're talking about, there could be 300,000 deaths of COVID-19 this year. Okay, so personally, I am strongly against the Swedish approach. I think it's the wrong way to do it. I think we have to have managed lockdowns in some respect.
Mm. So what global structures do you think, any of the panelists, are being put in place to stop these pandemics happening again? Or if they are inevitable, can we have a better strategy for how to deal with them?
Again, I think Eddie's probably best to answer that. In fact, did answer it at the end of your talk. That's what's required. What's the evidence that it's actually going to happen, Eddie?
Well, I think one of the most concerning things about it, I'm going to sort of relate to a previous point about this whole thing, is how political every act has become in this pandemic. Okay, so this thing is no surprise to me at all. This was going to happen, and the way it's played out is basically as you would predict. The one thing that has shocked me is simply how political everything is. Even simple things like wearing a face mask has been a political gesture. So here, what I think the concern to me is how WHO, who are the global structure that we have to do this, and who acted very early on and have given consistent advice all the way through, absolutely consistent advice at what to do. They've been, the US has now cut their funding off, and they're being, was pulled out of the WHO, and WHO is being accused of various playing political games. Now that's the concern. That is the concern. And I think to stop this happening in the future, we have to have global structures at least as strong as WHO was, if not stronger. Okay, and we can't freeze countries out, even though we may not get on with them politically. This is a global, it will only work if there's a global collaboration. We have to know what's going on in each country, and each country has to tell us. Otherwise, we will never ever stop these things. So I think we need to enhance WHO, and it's very concerning that they're being used as a political, as a target, because they're exactly what we need.
Hmm. So something that's also related in one of the other questions is, how do we then raise the importance of listening to the scientists and to the people who are actually producing evidence-based research? So you've already mentioned it becoming political. Is there some strategy that we should be all adopting to make the evidence and the importance of scientists and medical researchers more? Steve, is that your question?
I can make a start on that. I think by showing and doing, and being given the opportunity to show and do, and COVID has done that actually. I can't remember an occasion where there has been such global attention given to the emerging evidence and the science behind a problem. And it has been highly politicized, and it's been diverted and subverted, as Eddie says, but nonetheless, the value of science in this context has been both unarguable and demonstrable. So we need to build on this and to really take this as an example of what we need to do and how we can, as a sector, come together to start to really tackle some of these bigger other problems that are facing us.
Mm. So getting back to something more practical, a question that says, what are the most promising treatments for the virus as distinct from a vaccine? So what can we do sort of from now? I don't know, is that you or is that Eddie?
Oh, it's probably Eddie, actually. There's a really good, actually, on the ABC net today, there's a fantastically good account, I thought anyway, Eddie, largely as a layperson in the specifics of pharmaceutical and vaccine development. there's a really good account of the mechanisms that are being targeted and the way in which different antivirals and vaccines might work. It's on the ABC website today. I don't know whether you've seen that, Eddie.
No, I haven't. So there are for treatments there are a number of antivirals that that are on trial and they're in development. And what we'll probably need is a set of antivirals that attack different parts of our life cycle. That's the kind of best way of doing it, because then you reduce resistance. You've seen the results, some of the clinical trials so far. So things like remdesivir look pretty good for late stage patients. Hydroxychloroquine maybe not so. And these were mainly, I should say, these were mainly off the shelf antivirals they had prior to the outbreak that they've repurposed. But there's a huge, obviously, industry in developing these, but they will also need to go through for clinical trial and evaluation. So I don't think we have definitive evidence on any of those yet. So that will just have to come with enough cases. Probably an equally important thing is probably better managing clinical cases, particularly people who are on, in the ICUs. And treatments are getting there. We know that the inflammatory response is the kind of thing that is the most dangerous thing of all. And there are better medications out there now and treatments to kind of to control that. And it's very clear that we're handling the virus better now than we were in February and March. We now have a better understanding about actually how to do this. So, but like all these things, we can't. I know everyone's desperate to have, this is the thing that's got this magic bullet that's going to do it, and we're all going to release it, and but we need to go through the proper regulatory procedures, because there is a risk that like hydroxychloroquine, that we get excited about something that's actually dangerous, it turns out. So we have to wait for the clinical trials to take place, and they take time.
It's also the emerging evidence around the mechanisms of disease and damage caused by the virus, which, as Eddie said, are largely the response by our own bodies to the virus, and whether that be the inflammatory response or it's turning out that extensive capillary damage is occurring, and that's added to endothelial damage, and those things are causing extensive micro clotting, thrombosis throughout organs in the body, from brain to kidney to lungs and heart. So it isn't only a lung disease. And the way in which, as I was saying, some of the chronic disease conditions are actually exacerbating that. So we're seeing, for example, obesity and the deposition of fat within alveolar cells in the lung, so-called fatty lung, is dramatically increasing the risk of COVID infection. ACE2 receptors, which are the ones that the virus uses to get into our cells, how active they are, how dense they are in different tissues, and how that varies with different comorbidities is turning out to be important. So, and even things like the extent to which people have sleep apnea and are likely then to micro aspire saliva and with a load of virus deeper into the lung. These things are turning out to be potentially important as well. So managing our general health is more important than ever before, particularly as we age.
So I was going to say, Steve, to add on to that, one of the most sort of difficult things about this virus is it does affect so many tissue types. As you say, it's not just a lung infection. It affects your heart, brain, testes even. It's quite extraordinary. So it really has it's a very broad acting virus, which just makes it harder to control. And as you say, general health then becomes much more important.
So let me ask one more question to Ben, and then I think we'll start to wrap up with final questions. But one question first to Ben. So the current university structure, does that work well for this kind of multidisciplinary endeavour? I mean, or do we need to change the way universities are going to do their research?
Well, that's the big question of the day, isn't it? The post-COVID university vision And I guess, you know, that's a national conversation that we're having. I mean, at the University of Sydney, we're fortunate to have structures through these multidisciplinary initiatives that allow us to very efficiently, effectively, swiftly come together around some of these grand challenge projects. So I think what we've seen this year initially with the bushfire crisis, I think with the air quality issues that came out post-bushfire, and now with COVID, and many other areas, sustainability more generally, really swift response from within the university sector to bring researchers together across faculties and schools. But as I've always said, you know, schools, and you were a head of school, Anne, you were a wonderful head of school. Every school is a sovereign entity. Almost as if every academic is a sovereign entity. And, you know, the challenge we have, Steve and I have, and the other MDIs, and institutes that try to build multidisciplinary collaboration is the tension between the schools, the business units within the universities that have to put bums on seats and keep the business of the university going. And that's measured by that simple number of seats that are filled, with the institutes that are driving this large scale, big picture, societally relevant, grand challenge solving research, mission focused research, as Steve alluded to earlier. And I guess if you go back to the core values of an institution like the University of Sydney, and I keep saying this to my colleagues, we're not a business, we're not incorporated. We're really set up to achieve three things. One is to create knowledge, to inspire the next generation, and to engage with the community and have impact. And I think if we keep those three pillars front and center as we run these universities, I think that bodes well. But I think there are going to be some interesting conversations. We are going to need to reimagine, and we are reimagining university structures, because we're not going to be able to rely on the international student income for a number of years. And I'd like to think that the mission-focused research that has been so pivotal to the current crisis is front and center, and that we reach a nice balance of maintaining the disciplinary strengths, which are so vital to training, and we need to be anchored in that disciplinary strengths with a multidisciplinary mission-focused approach.
Thanks, Ben. And I'd like a last question to Eddie, and then I've got a final question for Steve. So for Eddie, are there ways in which Australia can be better prepared to respond to future pandemics? And how do we make people take things seriously?
I might leave that seriously thing maybe to Steve, because he's much more dealing with convincing people of various things. How Australia can do better? Australia's done well. Let's not get overly critical. We've done a very globally a very low number of infections, and the number of fatalities we've had is still relatively low. So that's good so far. I do think things could be better. I think what we're crying out for is a national CDC, Centre for Disease Control, like structure in this country. I am concerned that the way that different, not going to sort of, you know, knock the federation, but different states behave in different ways, and to me in a rather loose manner. I think, if only virtually, I do think a national centre that can coordinate and manage infectious disease outbreaks is really useful. And I would also say that centre, rather than just looking at human diseases, also has to integrate animal diseases, because what you're seeing here, this is what called a One Health thing. We cannot really distinguish infections in animals and infection in humans. They are part of the same thing now. Traditionally, we've split science into vets and medics, but that's a false dichotomy in many ways. Now we have to be more integrated. So I'd like to see a national framework that can manage human animal health and allow us have structures in place that we can act quicker and more in more targeted, smarter manner if these things happen again.
Thanks, Eddie. And Steve, last word for you, last question before we need to wrap up. Do we need to reconsider the way we train scientists and practice science to be more effective?
Yes, and I think I can echo both what Ben said, what Eddie's just said, and take us right back to the beginning of today. And that is, we need people to, of course, earn their spurs within their discipline. They need to have deep disciplinary understanding and expertise. They need to know the tools of their trade, but we need to train our young scientists to be able to work collaboratively across disciplines to look for the synergies that may be there that otherwise their blinkers would not allow them to see. And we need them to be able to engage with community better, to tell their stories in ways that the rest of society is inspired by and understands, and to listen to the rest of society. If we can do that, and it doesn't take too much more than instituting things like our multidisciplinary initiatives and encouraging through the structures in universities and in government, then we will have come a step change towards solving some of these really big, complex challenges.
Thank you. So thank you to the speakers. It's been a very interesting and informative session. And now I'd like to invite FTSE Fellow Dr. Peter Tyree to give a vote of thanks to our speakers. Peter's currently chairman of the Tyree Foundation, which has a long and distinguished record in the areas of education, community support and medical science. And Peter was recently made member of the Order of Australia for services to engineering and education. So you have a brief time, Peter, please.
Thank you very much, Anne. Before thanking our panel, I'd like to make the comment that I hope everyone has enjoyed this webinar today, our first joint webinar with FTSE and the Union University and Schools Club. The club has gone the extra yards by inviting a number of people to watch this video and have their usual sandwiches, which I hope we'll be going back to next month. Today, our panel has excelled. Each of us will have left this event today better off. We have a better understanding of COVID-19. We have a better idea of Australian research, how it is done, how it collaborates in local and global senses, and of course, recognising comments by Steve, we have gained first-hand visibility of three of Australia's very talented scientists and can see why Australia continues to build its worldwide image and reputation in scientific research. Steve, Eddie and Ben, we're indebted to you for making our wonderful contribution, your wonderful contributions today, and in doing so, helping many Australians on the road ahead in beating COVID-19. And dare I say, the importance of global collaboration. Before moving a vote of thanks, I want to advise that we'll be holding another event in late September, so stay tuned. To our three speakers and panellists, Steve, Eddie and Ben, at this time, please visualise well over 100 people standing and clapping in applause to thank you for your contributions and making the day a great success. Thank you very much.
Yeah. So thanks, Peter. And this now brings our webinar to a close. Thank you to all the panellists, the organisers and the technical staff at FTSE, and to all of you online for attending and for your questions. The final thing for me is just to say I wish you all continued health and safety in these uncertain times. So thank you and good afternoon.
Thank you.
Thank you.